Welcome to Sciencera. In this video, we're going to discuss the citric acid cycle, the central pathway of carbohydrates, lipids, and amino acid metabolism from chapter number 16 of Harper's Illustrated Biochemistry, edition 28. The objective for today's lesson is to see the reactions of citric acid cycle, how ATP is yielded. We're going to look at the vitamins used in citric acid cycle, what are the importance of the vitamins, catabolism of amino acid and route of their synthesis. Then we're going to look at the anaplerotic pathways that permit replenishment of citric acid cycle intermediate. Then we're going to look at the fatty acid synthesis, availability of oxidized cofactor, and then hyperammonia, which can also lead to the loss of consciousness. Let's begin. Biomedical importance of citric acid cycle. The citric acid cycle is a sequence of reaction in mitochondria that oxidize the acetyl moiety of acetyl coenzyme A to carbon dioxide and reduces coenzymes that are reoxidized through the electron transport chain linked to the formation of ATP. The next one is citric acid cycle is final common pathway for oxidation of carbohydrate, lipids and protein because glucose, fatty acid and most amino acids are metabolized to acetyl coenzyme A or intermediate of the cycle. It also has a central role in gluconeogenesis, lipogenesis and interconversion of amino acids. Many of these processes occur in the most tissue, but liver is the only tissue in which all occur to the significant extent. The few genetic defects of citric acid cycle enzyme that have been reported are associated with severe neurological damage as a result of very considerably impaired ATP formation in the central nervous system. Hyperammonia, as occurs in advanced liver disease, lead to loss of consciousness, coma, and convulsions as a result of impaired activity of citric acid cycle, leading to the reduced formation of ATP. Ammonia both depletes citric acid cycle intermediates by withdrawing the alpha ketoglutarate for the formation of glutamate and glutamine and inhibit the oxidative decarboxylation of alpha ketoglutarate. So these were the six important biomedical importance of citric acid cycle. The citric acid cycle provides the substrate for the respiratory chain. The acetyl moiety of acetyl coenzyme A react with 4-carbon dicarboxylic acid oxaloacetate to produce citrate, a 6-carbon tricarboxylic acid. Two carbon dioxide molecules are released and oxaloacetate is regenerated in succeeding process. Because oxaloacetate is regenerated at the end of the cycle, it can be regarded a catalytic component in oxidation of a substantial quantity of acetyl coenzyme A. The citric acid cycle provides substrate for the respiratory chain. The citric acid cycle is the primary mechanism to produce ATP from metabolic fuels that are oxidized. Coenzymes are reduced and then reoxidized in respiratory chain during the oxidation of acetyl coenzyme A, which is linked to the ATP production by oxidative phosphorylation. This is an aerobic process since the reduced coenzyme require oxygen as the final oxidant. Citric acid cycle enzymes are found in mitochondrial matrix, either free or linked to the inner mitochondrial membrane and the crista membrane together with the respiratory chain enzymes and coenzymes. The figure on the screen shows the citric acid cycle, the major catabolic pathway of acetyl coenzyme A. Acetyl coenzyme A, the product of carbohydrate, protein and lipid catabolism, enters the cycle by forming citrate and is oxidized to carbon dioxide with reduction of coenzymes. Reoxidation of the coenzymes in respiratory chain lead to phosphorylation of ADP to ATP. For 1 turn of the cycle, 9 ATP are generated via oxidative phosphorylation and 1 ATP or GTP arises at the substrate level from the conversion of succinyl coenzyme A to succinate. Reaction of citric acid cycle produce reducing equivalent of carbon dioxide. Let's see how this take place. Citrate synthase catalyzes the first interaction between acetyl coenzyme A and oxaloacetate to create citrate by forming a carbon-carbon bond between acetyl coenzyme A, methyl carbon and the carbonyl carbon of oxaloacetate. An exothermic process occurs when the thioester bond of the resulting citryl coenzyme is hydrolyzed, releasing the citrate and coenzyme A. The enzyme aconitase or aconitate hydratase isomerizes citrate into isocitrate in 2 steps: dehydration to cis-aconitate and rehydration to isocitrate. Even though citrate is a symmetrical molecule, aconitase react with it asymmetrically. Thus, the 2 carbon atoms that are lost in the cycle subsequent reaction are not the one that were added from the acetyl coenzyme A. This asymmetric behavior is caused by channeling, which is the direct passage of the product of citrate synthase to the active site of aconitase without passing through the free solution. This allows the citric acid cycle to work in tandem with availability of citrate in cytosol as an acetyl coenzyme A source for fatty acid production. Fluoroacetate is formed when fluorinated substances employed in anti-cancer treatment and industrial chemicals are metabolized. Because of fluoroacetyl coenzyme A condenses with oxaloacetate to generate fluorocitrate, which inhibits aconitase and cause citrate accumulation. It is hazardous. Isocitrate is dehydrogenated by isocitrate dehydrogenase to produce oxalosuccinate, which is then decarboxylated to alpha ketoglutarate. Mg2 plus or Mn2 plus ions are required for decarboxylation. Isocitrate dehydrogenase is divided into 3 isoenzymes. One is found only in mitochondria and utilizes nicotinamide adenine dinucleotide or NAD. The other two, which are present in mitochondria and cytoplasm, need NADP. The NAD plus dependent enzymes is involved in respiratory chain linked oxidation of isocitrate. Alpha ketoglutarate undergoes oxidative decarboxylation, which is catalyzed by a multi-enzyme complex identical to the one that catalyzes pyruvate oxidative decarboxylation. The alpha-ketoglutarate dehydrogenase complex produces succinyl coenzyme A by requiring the same cofactor as the pyruvate dehydrogenase complex. Thiamine diphosphate, lipotate, NAD plus, flavin adenine dinucleotide FAD and coenzyme A. This reaction's equilibrium is sufficiently skewed towards succinyl coenzyme A production that it must be deemed physiologically unidirectional. Arsenite slows the reaction like pyruvate oxidation, causing the substrate alpha ketoglutarate to accumulate. Ketoglutarate dehydrogenase is inhibited by high levels of ammonia. The enzyme succinate thiokinase converts succinyl coenzyme A to succinate. In the citric acid cycle, this is the only instance of the substrate level phosphorylation. The liver and kidney possesses two isoenzymes of succinate thiokinase, one specific for GDP and the other for ADP in tissue where gluconeogenesis occur. The GTP produced is employed in glucogenesis to decarboxylate oxaloacetate to phosphoenol pyruvate, and it serves as a regulatory link between citric acid cycle activity and oxaloacetate withdrawal for gluconeogenesis. Only the isoenzymes that phosphorylates ADP is found in non-gluconeogenic tissues. When ketone bodies are being metabolized in extrahepatic tissue, there is an alternative reaction catalyzed by succinyl coenzyme A, acetoacetate coenzyme A transferase involving the transfer of coenzyme A from succinyl coenzyme A to acetoacetate forming acetoacetyl coenzyme A and succinate. The onward metabolism of succinate leading to the regeneration of oxaloacetate is same sequence of chemical reaction as occur in the beta oxidation of fatty acid. Dehydrogenation to form a carbon-carbon bond, addition of water to form a hydroxyl group, and further dehydrogenation to yield the oxo group of oxaloacetate. First dehydrogenation reaction forming fumarate is catalyzed by succinate dehydrogenase, which is bound to the inner surface of the inner mitochondrial membrane. The enzyme contains FAD and iron sulfur FeS protein and directly reduces the ubiquinin in the electron transport chain. Fumarase or fumarate hydratase catalyzes the addition of waters across the double bond of fumarate, yielding malate. As we said earlier, fumarase catalyzes the addition of water across fumarate's double bond to produce malate. Malate dehydrogenase reduces NAD plus by oxidizing malate to the oxaloacetate. Even though the equilibrium of this reaction favor malate, the net flux is to oxaloacetate due to the constant removal of oxaloacetate and the constant reoxidation of NADH. Here you can see the citric acid cycle. We have acetyl coenzyme A, then we have citrate, cis-aconitase, isocitrate, oxalosuccinate, alpha-ketoglutarate, succinyl coenzyme A, succinate, fumarase, L-maltate, and then we have oxaloacetate. The mnemonic for citric acid cycle can be Cindy is kinky so she fornicates more often. C for citrate, I for isocitrate, K for alpha ketoglutarate, S for succinyl coenzyme A, S for succinate, F for fumarate, M for malate, and O for oxaloacetate. 10 ATPs are formed per turn of the citric acid cycle. As a result of oxidation catalyzed by the dehydrogenase of citric acid cycle, 3 molecules of NADH and 1 of FADH are produced for each molecule of acetyl coenzyme A catabolized in 1 turn of the cycle. These reducing equivalent are transferred to the respiratory chain where reoxidation of each NADH results in the formation of 2.5 ATP and of FADH2, which is 1.5 ATP. In addition, 1 ATP or GTP is formed by substrate level phosphorylation catalyzed by succinate thiokinase. In the table given, you can see the reaction with isocitrate dehydrogenase produce the NADH, which is equal to the 2.5 ATP. Alpha ketoglutarate dehydrogenase also produce NADH, which is equal to 2.5 ATP. Succinate thiokinase at the substrate level produce 1 ATP or GTP. Succinate dehydrogenase produce FADH2, which is equal to 1.5 ATP. Malate dehydrogenase produce NADH, which is equal to 2.5. If you add them together, you get 10 number of ATPs produced per acetyl coenzyme A. Vitamins play a key role in citric acid cycle. Four of the B vitamins are essential in citric acid cycle and hence energy yielding metabolism. Riboflavin, also known as vitamin B2 as FAD, is the cofactor for succinate dehydrogenase. Niacin vitamin B3 as NAD plus is the electron acceptor for isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase and malate dehydrogenase. Thiamine vitamin B1 as thiamine diphosphate is the coenzyme for decarboxylation in alpha ketoglutarate dehydrogenase reaction. And pantothenic acid or vitamin B5 as part of the coenzyme A is esterified to carboxylic acid to form acetyl coenzyme A and succinyl coenzyme A. So four vitamins are vitamin B2, vitamin B3, B1 and vitamin B5. The citric acid cycle plays a pivotal role in metabolism. The citric acid cycle is not only a pathway for oxidation of two carbon unit but it is also a major pathway for interconversion of metabolite arising from transamination and deamination of amino acid and providing the substrate for amino acid synthesis by transamination as well as for the gluconeogenesis and fatty acid synthesis because it functions in both oxidative and synthetic processes. It is amphibolic in nature. The citric acid cycle takes part in gluconeogenesis, transamination, and deamination. All the intermediates of cycle are potentially glucogenic since they can give rise to oxaloacetate and hence production of glucose in liver and kidney which carry out gluconeogenesis. The key enzyme that catalyzes transfer out of the cycle into gluconeogenesis is phosphoenol pyruvate carboxykinase which catalyzes the decarboxylation of oxaloacetate to phosphoenol pyruvate with GTP acting as a phosphate donor. Among the most important of such an anaplerotic reaction is formation of oxaloacetate by carboxylation of pyruvate catalyzed by the pyruvate carboxylase. This reaction is important in maintaining an adequate concentration of oxaloacetate for condensation reaction with acetyl coenzyme A. Lactate, an important substrate for gluconeogenesis, enters the cycle via oxidation to pyruvate and then carboxylation to the oxaloacetate. Glutamate and glutamine are important anaplerotic substrate because they yield alpha ketoglutarate as a result of the reaction catalyzed by the glutaminase and glutamate dehydrogenase. Transamination of aspartate leads directly to the formation of oxaloacetate and a variety of compounds that are metabolized to yield propionyl coenzyme A which can be carboxylated and isomerized to succinyl coenzyme A are also the important anaplerotic substrate. Because these reactions are reversible, the cycle also serves as the source of carbon skeleton for the synthesis of these amino acids. Other amino acids contribute to the glucogenesis because their carbon skeleton give rise to the citric acid cycle intermediate. The citric acid cycle itself does not provide a pathway for the complete oxidation of the carbon skeleton of amino acid that give rise to the intermediates such as alpha ketoglutarate, succinyl coenzyme A, fumarate and oxaloacetate because this results in an increase in amount of oxaloacetate. For complete oxidation to occur, oxaloacetate must undergo phosphorylation and carboxylation to phosphoenol pyruvate, then dephosphorylation to pyruvate, and oxidative decarboxylation to acetyl coenzyme A catalyzed by pyruvate dehydrogenase. In ruminant whose main metabolic fuel is short chain fatty acid formed by bacterial fermentation, the conversion of propionate, the major glucogenic the major glucogenic product of rumen fermentation to succinyl coenzyme A via the methyl malonyl coenzyme A pathway is especially important. The citric acid cycle takes part in fatty acid synthesis. Acetyl coenzyme A formed from the pyruvate by action of pyruvate dehydrogenase. Pyruvate dehydrogenase is mitochondrial enzyme and fatty acid synthesis is a systolic pathway. The mitochondrial membrane is impermeable to acetyl coenzyme A. For acetyl coenzyme A to be available in cytosol, citrate is transported from the mitochondrion to the cytosol, then cleaved in a reaction catalyzed by the citrate lyase. Citrate is only available for transport out of the mitochondrion when aconitase is inhibited by its product and therefore saturated with its substrate, so that citrate cannot be channeled directly from citrate synthase onto the aconitase. This ensures that citrate is used for fatty acid synthesis only when there is an adequate amount to ensure the continued activity of the cycle. The oxaloacetate released by citrate lyase cannot re-enter the mitochondrion but is reduced to malate at the expense of NADH, and the malate undergoes oxidative carboxylation to pyruvate, reducing NADP plus to NADPH. This reaction, catalyzed by the malic enzyme, is the source of half the NADPH required for fatty acid synthesis. The remainder is provided by the pentose phosphate pathway. Pyruvate enters the mitochondrion and is carboxylated to oxaloacetate by pyruvate carboxylase, an ATP-dependent reaction in which the coenzyme is vitamin biotin, vitamin B7 or vitamin H. Regulation of citric acid cycle depends primarily on a supply of oxidized cofactor. In most tissue where the primary role of citric acid cycle is the energy yielding metabolism, respiratory control via the respiratory chain and oxidative phosphorylation regulates the citric acid cycle activity. Thus, activity is immediately dependent on the supply of NAD+, which in turn, because of the tight coupling between oxidation and phosphorylation, is dependent on the availability of ADP and hence ultimately on utilization of ATP in chemical and physical work. The main site for regulation are non-equilibrium reaction catalyzed by pyruvate dehydrogenase, citrate synthase, isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase. Succinate dehydrogenase is inhibited by oxaloacetate and the availability of oxaloacetate is controlled by malate dehydrogenase and dependent on the NADH to NAD plus ratio. This is largely because of the withdrawal of alpha-ketoglutarate to form glutamate and then glutamine, leading to lowered concentration of all the citric acid cycle intermediate and hence reduced generation of ATP. The equilibrium of the glutamate dehydrogenase is finely poised and the direction of reaction depends on the ratio of NAD plus, NADH and then the concentration of ammonium ion. This brings us to the end of the citric acid cycle. Let's look at the summary. The citric acid cycle is the final pathway for oxidation of carbohydrates, lipid and protein. Their common end metabolite, acetyl coenzyme A, reacts with oxaloacetate to form citrate. By a series of dehydrogenations and decarboxylation, citrate is degraded, reducing coenzymes, releasing 2 carbon dioxide and regenerating oxaloacetate. Then reduced coenzyme are oxidized by a respiratory chain linked to the formation of ATP. Thus, the cycle is the major pathway for the formation of ATP and is located in the matrix of mitochondria adjacent to the enzyme of the respiratory chain and oxidative phosphorylation. The citric acid cycle is amphibolic since in addition to the oxidation, it is important in provision of carbon skeleton for gluconeogenesis, acetyl coenzyme A for fatty acid synthesis and interconversion of amino acid. Thank you for watching. If you like my video, don't forget to subscribe for more.